3604 Industrial Biotechnology. In this lecture, we will study about fermentation. So first of all, let's see what is a fermentation. Fermentation is a metabolic process in which an organism converts carbohydrate, for example starch or glucose, into alcohol or an acid. The industrial microorganisms are normally cultivated under rigorous control condition developed to optimize the growth of the microorganism. So the microorganism that we are using our industry are normally optimized in a way to produce much larger product. The synthesis of microbial metabolite is usually tightly regulated by the microbial cell. So the number of microbial cell actually depends upon the number of metabolite that are produced. For example, if there are a large number of microorganism and microbial cell, then ultimately a large number of metabolite will be produced. Fermentation are performed in large filters or fermenters that we know with the capacity of several thousand liters. These are maybe simple tanks which may be stirred or unstirred, that we will study in our next chapters. Complex integrated system involving various level of computer controls. The fermenter material should have the following characteristics. So the fermenter that we are using must have the following characters in order to obtain a purified and sufficient amount of end product. That must be usually stainless steel in order to avoid contamination, can be repeatedly sterilized, will not react adversely with the microorganisms. So the microorganisms that we are using in this fermenter must not react with the fermenter, will not react to the target product, and the end target product that is produced must not react to the fermenter as well. Major influence on fermentation performance are the mode of fermenter operation. For example, either we are using batch for batch or fed batch or continuous system. So all these systems we will study in the future lectures. The method of its aeration and agitation. Aeration is the process by which air or oxygen is provided to the microorganism, while agitation is the stirring of this the initial fermenter product. Approach taken to process scale up. So here we can see there are two processes actually, upstream processing and downstream processing. What happens in the upstream processing is the fermentation raw material are used as the input and microorganism of the desired character are produced, and then the fermentation take place. When the fermentation takes place, then the downstream processing starts. In the downstream processing, what we do is the product that are produced is purified and we get the desirable end product that is ready to use. And if we have a sufficient amount of waste product, that is again used in the fermentation as a fermentation raw material in the fermentation process.